Bueno, bon dia a tothom. Estem aquí reunits perquè això és l'acte de defensa de la tesi doctoral que presentarà l'Alba Ventós, que porta per títol Factors, que poso els ulleres si no ho veuré, Factors Determining the Adult Body Plan in the Metabolic Embryogenesis. Una tesi que està feta a l'Institut de Biologia Evolutiva, un centre mixt entre el CSIC i la UPF, i dirigida pel doctor Xavier Vallès, que està present aquí. Ara farem dues fases. La primera fase és l'exposició, després presentarem les conclusions i després la fase de defensa. I per la primera fase de l'exposició tens uns 45-50 minuts. El tribunal està format per la doctora Isabel Navarro, de la Universitat de Barcelona, la doctora de l'Hostiolacs, del CSIC, del Centre de Biologia Evolutiva, i jo mateix, Francesc Piferret, de l'Institut de Ciències del Mar. Gràcies per la vostra assistència, sense més preàmbul, podem donar per començar aquesta defensa. Aquesta exposició.
És molt complicat, això.
Tranquil·la.
Hem d'apagar els telèfons mòbils, si pot ser, perquè no hi ha interrupcions.
Això es pot tancar, no?
Sí.
So, hello and thank you for the introduction. I'm Alba and today I'm defending my PhD thesis, which is entitled Factors Determining the Adult Body Plan in the Hemimetabolan Embryogenesis. So, this is the outline that I will follow during my presentation. I will start with a brief introduction, then I will tell you about my hypothesis, then I will present you my results, and then the conclusions. So, without further delays, let's start with the introduction. Insects are 1 million species formally described, and they represent the most diverse animal group, not only in their number of species, but also in their morphological, physiological, and lifestyle diversity. This is due in great part to their long evolutionary history. During their evolutionary history, two key innovations happen. One of them is the appearance of functional wings, and with that, powered flight, and the other one is the appearance of metamorphosis. Based on the insect development, we can classify insects in three groups. Primitive wingless insects that we call ametabolan, and these are without wings. And then we have two groups with wings, which are hemimetabolan insects and holometabolan insects. The ametabolan insects, doesn't have wings, when they hatch, they already have similar morphology and lifestyle than the adult, and they continue molting during their whole lives. Even they have more than one sexually mature instar. If we go to the hemimetabolan insects, when they hatch, they have also the same morphology and lifestyle than the adult, but when they reach the adult stage, they don't molt anymore, having only one sexually mature instar. So, to the transition from the last nymphal instar to the adult, we call that metamorphosis. And then, during this transition, is when the animal acquires the functional genitalia and the wings. Then we have holometabolan insects. These ones, when they hatch, they have a vermiform shape, very different from the adult. And they need a intermediate stage called pupa to reach the morphological gap between the larvae and the adult. Nowadays, the most established hypothesis is that holometabolans originated from hemimetabolan species. And most studies regarding this are focusing on post-embryonic development. But as you can see, both modes of metamorphosis, the differences between them start at embryogenesis, because hemimetabolan embryogenesis gives rise to a nymph which is already similar to the adult, while holometabolan embryogenesis gives rise to a larvae which is different from the adult. So from here, it came my main hypothesis, that is that there has to be something in the embryo that determines which kind of metamorphosis will take place. So, in order to answer my main question, which is how holometabolan embryogenesis diverged from hemimetabolan, giving rise to a kind of worm instead of a miniature adult, we did different analyses. First of all, what we did in the lab is to compare two different transcriptomic data sets. One of holometabolan insects, which is Drosophila melanogaster, and another one of hemimetabolan insects that we take Blatella as a model. And then comparing the behavior of different genes in these two models, we select those that are behaving differently, so can be candidates for determining the mode of metamorphosis. Doing so, we selected 3 candidates. One of them is DNA methyltransferase 1. DNA methyltransferase 1 is a DNA methylase that methylates DNA, okay, which is adding a methyl group into DNA. What we found is that it is present during Blatella germanica embryo, but it is not present in Drosophila. Another candidate is Zelda. Zelda is a key factor during the maternal to zygotic transition. This maternal to zygotic transition is when maternal genes start to be degraded and zygotic genes start to be expressed. So Zelda has been described to be a key factor during this transition in Drosophila, and what we found is that in Blatella, it is expressed in a huge peak at early embryogenesis, but it is present during the whole embryogenesis of Drosophila. So this different behavior made us thought that maybe Zelda is acting differently in both modes. Then we have E93. E93 is a factor that has been studied during post-embryonic development and is known to be determining the adult morphogenesis. So what we found is that it is present during the embryogenesis of Blatella, but in holometabolan insects, concretely in Drosophila, it is not present during the embryogenesis. So we thought that maybe E93 is activating the adult morphogenesis in the embryo. So let's go to the results. First of all, I will talk about DNMT1. Remember, DNMT1 is a DNA methyltransferase which methylates DNA, and it was peaking at early embryogenesis in Blatella, and it is not present in Drosophila. So what we wanted to answer during this work was, is DNMT1 promoting DNA methylation in Blatella? Is DNA methylation related to embryo development? Is DNA methylation related with gene expression? And how general is the role of DNMT1 in insect embryogenesis? So, in order to answer these 4 questions, we first look for all DNMTs that we can find in Blatella's genome, and we find 2. One is DNMT1, and the other one is DNMT3. Both of them contain all the characteristic domains that are described not only for insects but also for other vertebrates or even humans. And then we check the expression during the embryogenesis of Blatella. And what we observe is that both are peaking at early embryogenesis of Blatella. Moreover, if you check the levels of expression, you see that DNMT1 is 100 times more expressed than DNMT3. Then we tried to know the function of these DNMTs. So to do so, we did maternal RNAi, which consists on injecting a double strand of RNA to the adult mother and see what happens to the embryo. What we observed is that we could deplete DNMT1, but we couldn't deplete DNMT3. We think that this is because of the low levels of expression of DNMT3. So we decided to continue our studies focusing only on DNMT1. When we did the maternal RNAi, what we expect of a control animal is that the adult female carried the ootheca with all the eggs, developing the embryos during 18 days. In treated animals, what happened is that after 18 days, embryos couldn't hatch. So what we did is to wait 2 days more and then open to see what was happening to those embryos. And what we observed is that the major phenotype was this one, which is a white yolk mass. This means that the embryos stop developing at early stages of development. So what we decided is to open the embryos before the OCK before to see what was happening to the embryos just after the peak of expression of DNMT1. So we opened at day 4. What we observe here is that most of the embryos were showing this phenotype, which means that the embryos couldn't develop beyond year-month formation. Then, in order to know if DNMT1 was methylating DNA in Blatella, we did a technique called reduced representation bisulfite sequencing, or RRBS. This technique consists on enriching your sample in CG-rich regions and then analyzing the levels of methylation of each region. What we saw is that in control embryos, methylation is higher towards 3' UTR, and also is higher in intergenic genes and exonic genes. Moreover, when we compare control embryos with treated embryos, we see that the DNA methylation levels were reduced in all regions. Then we wanted to know if those methylated genes have any role or function, and we did an AGO enrichment analysis, and what we observed is that methylated genes were enriched in function related with metabolic processes, neurogenesis, or cytosolic transport. And methylated genes were enriched in signaling pathways and leg patterning. Then another question that we have that we thought it was interesting is if the methylation regulates somehow the expression of the genes. And we did different analyses. The first one was to check the levels of expression of different genes at different stages of development and comparing methylated genes versus non-methylated genes. What we observed is that in all cases, methylated genes show higher levels of expression than non-methylated genes. And this is even more clear during the first period of embryogenesis. And this also happens independently on the gene region that is methylated. Then another thing that we wanted to know was if the expression changed more or less in those genes that are methylated. In order to do so, we compare the expression levels, or we calculate the expression change between embryo day 2 and embryo day 4, and then we compare this expression change between methylated and unmethylated genes. And what we observe is that methylated genes show less expression change regardless of if it was incremental or decremental, and also less expression variance. And then we wanted to know if this change in expression was also between replicates. So what we did is to compare the coefficients of variation of 2 different replicates for a same time point, and then we analyzed, we compare methylated versus non-methylated genes. What we observed is that methylated genes show less expression variability between biological replicates. And again, this is more evident at the first period of embryogenesis, which is where DNMT1 has its peak of expression. So, answering my first questions of this chapter, DNMT1 is promoting DNA methylation, as observed in other insect species. DNA methylation seems to be related with embryo development, especially during germ band formation. DNA methylation promotes, higher levels of DNA methylation correlated with higher expression levels, lower expression change, and less transcriptional noise. And then we know that DNMT1 is key for the embryogenesis for different species, but the current evidence and bibliography shows that the specific point of action of DNMT1 during the embryogenesis of the different species is different. So we can say that DNMT1 is instrumental for embryogenesis in cockroaches to mammals, but the current evidence indicates that the specific action varies in different lineages, even within insects. So let's move to my second chapter. I will drink a bit of water, please. Okay, so the second chapter is regarding Zelda. Zelda, remember, is a key factor during the maternal to zygotic transition, at least in Drosophila. And what we observe is that Zelda is expressed during the early embryogenesis in Platella, while it is expressed during the whole embryogenesis in Drosophila. So the questions that we have in this chapter were: is Zelda necessary for embryo development of Platella? Has Zelda any role during the maternal to zygotic transition also in Platella? Has Zelda any role during post-embryonic development in Platella? Might Zelda be involved in evolution to holometaboly? In order to answer this, we first check that our qRT-PCR data corresponds to our transcriptomic data, and we can see that both of them show a huge peak of expression of Zelda at the beginning of embryogenesis, and then we compare this data with the transcriptomic data of Drosophila and Tribolium, both of them showing expression during the entire embryogenesis. Moreover, we wanted to know where is Zelda expressing, and for that we did in situ hybridization at embryo day 3, and we observed that Zelda is expressed in the abdominal growth zone of Platella. Then we repeated the same approach, and like in the last chapter, we did maternal RNAi, and we observed what happens to the embryo. What we observed is that most of them show these kind of phenotypes, which regards the formation of the abdomen. In order to see that better, we decided to open again at embryo day 4, and see what was happening there to the embryo. Again, we choose embryo day 4 because it's after the peak of expression of Zelda. And what we observed is that most of them were showing these phenotypes, which means that the embryo couldn't segmentate well. Then, to know if Zelda has any function related with post-embryonic development, we did an RNAi, but this time during post-embryo development, injecting in NIM5 and see what was happening, if the insect can molt properly or not. And what we observed is that we didn't have any phenotypic effect. Then, to know which genes are regulated by Zelda, we analyzed different set of genes. First of all, we analyzed the NNT1, and the NNT1 was downregulated. Then we analyzed another set of microRNAs, which is microRNA 309. This family is known to be degrading mRNA during the maternal to zygotic transition in Drosophila. So we thought that if Zelda is a key factor during this transition also in Platella, it might be activating MIR309. And as you can see, levels of MIR309 were lower, so it means that Zelda is activating MIR309. Then, since most of the phenotypes that we observed were regarding segmentation, we analyzed some segmentation genes. We analyzed different maternal genes, gap genes, pair rule, and segment polarity genes. And most of them were downregulated in Zelda treatments. So, for this chapter, we could say that Zelda is essential for embryo development, especially during abdominal elongation. Zelda has a role in the maternal to zygotic transition, both activating the zygotic genome and activating MIR309, so activating the degradation of mRNAs, maternal mRNAs. Then, Zelda has any role in post-embryonic development, and maybe the narrow peak of Zelda at early embryogenesis of Platella is associated with the formation of a nymph instead of a larvae. Then let's go for the last chapter, which is regarding E93. E93, remember, has been studied in post-embryonic development, and it is known to be the adult specifier. It promotes the adult morphogenesis. And it was present in Platella embryo, but it is not present during Drosophila embryogenesis. So, in order to go to this chapter, we wanted to answer these questions. First, is E93 necessary for embryo development in Platella? Has E93 any role during the maternal to zygotic transition in Platella? Is the expression of E93 in the embryo an exclusive feature of hemimetabolan species? And might E93 be involved in the evolution of holometaboly? Again, in order to answer this, what we first did is to analyze the expression pattern of E93 in the embryo. So what we observe is that it is present in the transcriptomic data, but when we validate by qRT-PCR, we observe that E93 peaks at early embryogenesis, especially at day 1 of embryogenesis. So we did a maternal RNAi, to deplete E93, and we observed that we could deplete it. Then we observed the embryos, and we observed that most of them were showing this phenotype, which again corresponds to an embryo that couldn't develop beyond early stages of development. Then we decided to open before, just after the peak of expression of E93. So we opened at embryo day 3. Normally, at embryo day 2 or day 3, we have these dots that corresponds to energies that migrate to the surface of the embryo, formatting this line. But what we observed is that most of them were showing this phenotype, which means that the energies are formatting aggregates are not migrating to the surface of the egg, meaning that the embryos stop developing before forming the germ band. In order to know which genes are regulated by E93, we analyzed different set of genes. The first of them were some genes that are expressed just after the peak of expression of E93, so at embryo day 2, and we compared control and treated embryos. As you can see, treated embryos show lower levels of expression of these genes than control embryos. That means that these genes were not activated in E93 depleted embryos. Then we analyzed some genes that normally vanish after the peak of expression of E93, so vanish at embryo day 2. When we compare the expression levels of control embryos and treated embryos at this stage, we see that treated embryos show higher levels of expression than control embryos, meaning that maternal genes didn't vanish at embryo day 2 in E93 depleted embryos. So, as I told you before, methylphenidate is in charge of degrading those maternal mRNAs during the maternal to zygotic transition. So, may this methylphenidate be involved to these don't vanish mRNAs? So, let me introduce you to methylphenidate in Blatella. Methylphenidate in Blatella has a primary transcript that produces 4 precursors, A, B, and C, and D, and each of these precursors produces one mature sequence. But B and C precursors produce the same mature sequence, which we call P. As you can see, these 3 mature sequences, these 2 are very similar, but this one is very different to the 2 of them. And if you compare the sequence of these 2 versus the sequence of other insects, they are more conserved. So what we decided is to focus our study only in these 2 microRNAs. What we did is to first analyze the expression pattern of these microRNAs. We studied the precursors because we have the transcriptomic data. And then we saw that they peak at embryo day 2. Then we wanted to know if these microRNAs were downregulated in E93 depleted embryos. And what we observed is that they show less levels of expression in treated embryos. So we know now that E93 is promoting the production of methylphenidate mRNAs, but do these microRNAs regulate the decline of maternal mRNAs? In order to know that, we choose a maternal mRNA as a candidate. We choose Zelda because, you know, I was studying Zelda in other chapters, so it was a good candidate. And we use different algorithms like Starmie, Miranda, PITA, and these algorithms, and we predicted 6 sites at the 3' UTR of Zelda. Then we validate these 6 sites doing a reporter assay. This reporter assay was a luciferase assay which consists on cloning at the 3' UTR of a luciferase the sequence of your target sequence of the 3' UTR, and then co-transfect this with your microRNA into S2 cells. Here you can have 2 cases. First case is that your microRNA doesn't bind your site, and then the translation goes okay, so you have luciferase and you have light. The second option is that your microRNA binds the site, so there is a cleavage of mRNA, the luciferase is not produced, and you don't have light. Doing so, we obtained 2 positive sites that were active sites for methylphenidate microRNA at Zelda. So we can say that methylphenidate could bind Zelda at least in vitro. So taking all this together, we can build this model where E93 and Zelda are activating the zygotic genome. Both of them are activating the expression of methylphenidate A. that it at the same time is repressing cell expression, so making this peak that huge at early embryogenesis. So now the question is that an exclusive feature of hemimetabolans or of Blatella, or is something of general stuff of hemimetabolan insects? So we compare different hemimetabolan insects and different olometabolan insects, and what we observe is that in all hemimetabolan insects, E93 was present during embryogenesis, while in olometabolan insects it is not present at all, or very low levels. So it seems to be something exclusive of all hemimetabolan insects. So answering my questions, it seems like E93 is necessary for embryo development, especially for germ formation. E93 activates the zygotic genome expression, including methylphenidate and directly downregulating cell. The expression of E93 seems to be exclusive of hemimetabolans, but of course we will need to add more animals to the study because we only studied 5. And the last question, might E93 be involved in the evolution of olometabolism? Let me first compare these two models. The one that we built is this one, where E93 will be implementing the NIFA genetic program, and Zelda will be activating the zygotic genome, but only for abdomen elongation. Then, if we go to the bibliography that we have in Drosophila, Zelda will be activating the zygotic genome, in this case acquiring more functions because it's expressed during the whole embryogenesis, and activating the larval genetic program. So the output will be different. So, if E93 determines the NIFA genetic program in the hemimetabolan embryo, it could be that the loss of expression of these genes in the olometabolan embryo has facilitated the implementation of a larval genetic program. And that's the evolution of olometabolism. So, taking all this together, and taking my first question of the main question of my thesis, in order to evolve from here to here, 3 changes were needed. First, the loss of E93 expression, and with that, the loss of NIFA genetic program, the lower DNA methylation, and the expansion of cell expression, and the change on its function, and of course other changes. So, if now the committee agrees, I can proceed to read my conclusions. Yes? Thank you. So, I have 10 conclusions. 1. Depletion of DNMT1 resulted in a reduction of DNA methylation levels in the genome and impaired embryo development. This indicates that DNMT1 promotes DNA methylation in Blatella as observed in other insect species, and that this DNA methylation is necessary for proper embryo development. Methylated genes showed higher levels of expression than unmethylated genes, and GO enrichment analysis revealed that methylated genes are involved in metabolic processes, while unmethylated genes are more related with signaling pathways. This indicates that DNA methylation influences the level of expression of the genes, maintaining highly expressed those genes with housekeeping functions, as shown in other insect species. High levels of DNA methylation correlated with low expression variability, both between biological replicates and between different developmental stages. This supports the hypothesis positing that DNA methylation in gene regions reduces transcriptional noise, as previously suggested by authors working on human tissues. Cell depletion impaired embryo development, mainly involving different degrees of abdomen malformation. This indicates that cell is required for proper embryogenesis, in particular regarding the formation and elongation of the abdomen. Expression of early zygotic genes and methylphenidate microRNAs was downregulated in cell-depleted embryos. These observations indicate that cell induces the activation of the zygotic genome, including the production of methylphenidate microRNAs in the maternal to zygotic transition context. In Blatella germanica, cell expression concentrates in early embryogenesis during the maternal to zygotic transition, while in Drosophila melanogaster and Tribolium castaneum, it continues beyond maternal to zygotic transition. This suggests that the mechanism regulating cell temporal expression is different in the hemimetabolan and olometabolan species, and that the narrow peak of Zelda at the beginning of embryogenesis of Platella may be associated with the formation of the adultoid nymph, whereas its extension in holometabolous species may be associated with the formation of a derived larva. Depletion of E93 impaired embryo development before germ formation and triggered a number of malformations in embryos that pass that stage, including the formation of individuals with a vermiform shape. The whole data suggests that E93 is crucial for the germ formation of the adultoid nymph. E93 depleted embryos showed early zygotic genes and myrcene microRNAs downregulated. This indicates that E93 activates the zygotic genome, including the production of myrcene microRNAs, thus having a key role in the maternal to zygotic transition. The RNAi experiments showed that E93 contributes to the increase of myrcene microRNA levels and to the decline of Zelda expression in early embryogenesis. Moreover, the luciferase assays demonstrated that Zelda mRNA contains functional myrcene A target sites. This evidence suggests that E93 indirectly downregulates Zelda expression by enhancing the production of myrcene A. E93 is consistently expressed in early embryogenesis of Platella, which is not the case in Drosophila melanogaster. The data available in other species suggests that this could be a general role in hemimetabolans and non-metabolans, respectively. If E93 determines the nymphal genetic program in the hemimetabolan embryo, it could be that the loss of expression of this gene in the holometabolan embryo has facilitated the implementation of a larval genetic program and the evolution of holometaboly. And with that, thank you for your attention.
Thank you very much. Ara, com és preceptiu, passarem a la fase de defensa de les tècniques de l'exposició. I volia preguntar si hi ha alguna alçada que no entenc de l'anglès. Esperem en castellà. Endavant, doncs,